This is part 5 of the chapter 9, section A lecture notes, and I'm hoping this will be the last part because we don't have that many slides left. We left off on this slide where we were talking about what happens when the action potential is propagated down the T tubules, and that leads to a release of calcium ions from the sarcoplasmic reticulum. Now, what the calcium ion is going to do is it may be easier if I just draw this and if you draw it as well along with me. I'm not going to draw both strands of the actin. I'm just going to draw one strand because it's a lot to draw. But we see that the actin kind of forms something that looks like beads, like a string of beads or string of pearls. And I'm going to put a dot in the center of each, which is going to represent the active site on the actin where the myosin is going to bind. So what happens is you have something preventing myosin from binding. And I'll do that in a different color. And see, even if you don't draw well like me, I don't understand it. Okay, so even if you don't draw well like me, you can still draw and get the idea of what's going on. So there is a molecule that I'm going to draw now called tropomyosin, which is basically covering up these active sites. So if myosin can't bind, then you can't have a muscle contraction. So what is holding the tropomyosin in place is another molecule called troponin. And we'll do that one in a different color. You usually see it in yellow. It kind of looks like an upside down snowman. All right, so this is what the calcium does. The calcium moves the troponin. I know it's hard to see this, but color I used. The calcium moves the troponin out of the way, which lets the tropomyosin uncover those active sites. And once those active sites are uncovered, myosin filaments are able to attach to the active sites on the actin, and a contraction can then occur. So the calcium ion, the role of the calcium ion is to come in and bind to the troponin. Go back to red. The calcium ion comes in and binds to the troponin. That troponin changes and moves, changes shape or something, and it allows the tropomyosin, which is covering the active sites, to move out of the way. Now the active sites are exposed and myosin can bind to them. When calcium concentration is low, tropomyosin blocks the active sites on actin because the tropomyosin is being held in place by troponin. So let's add that to this note. The tropomyosin is covering the active sites, but what is holding the tropomyosin in place is troponin. Okay, the myosin heads cannot attach to the actin, so what happens is the voltage sensitive proteins in the T tubules change shape, and they cause the sarcoplasmic reticulum to release calcium ions into the cytosol. So in other words, when we have the sarcolemma of the muscle fiber, kind of folding in and forming this T tubule, the action potential is going to travel down the sarcolemma Remember, it was negative, so it's going to change to positive, and it's going to propagate all the way down the T-tubule deep into the muscle fiber, and this will trigger the release of those calcium ions. And then the calcium binds to the troponin. Troponin causes the tropomyosin to move away from the binding sites on actin. The myosin heads then attach to the actin, forming a cross bridge. So this is cross bridge formation. And once that happens, once the myosin is attached to the actin, it can pull the actin filaments toward each other, and that causes a shortening of the sarcomere. And then the calcium ions are pumped back into the sarcoplasmic reticulum so they can be reused. So here are the 4 steps of the cross-bridge cycle. Cross-bridge formation, that's when the myosin attaches to the active sites on actin. And then the power stroke is when the myosin pulls the actin filaments toward each other, toward the end line. And then what allows for the myosin to unattach or detach from the actin is ATP. When ATP attaches to the myosin head, it causes the myosin to unattach from the actin. And that's going to put the myosin in a high energy state to be used for the next power stroke. All right, again, the events of the cross-bridge cycle. This is showing you myosin attached to actin. So this is the forming of the cross bridge, and that happens because calcium is released from the sarcoplasmic reticulum. It causes the troponin to change shape, and the tropomyosin, which is yellow here, moves out of the way, and the active sites on actin are exposed. You can see them exposed here and here, and the myosin heads will attach, will come up and attach to those active sites on actin. And then you will have the power stroke. See how the myosin is at this angle, and then it kind of moves that way. So it moves, it pulls the actin filaments toward each other. And ATP will then attach to the myosin head, and that will cause it to unattach or detach from the actin, and now it can be in position to form another cross bridge. Now, we've heard the term rigor mortis, which occurs 3 to 4 hours after death when the body begins to stiffen. Now, after after a certain time, the body doesn't stay that way. It stays that way for a certain time, and then it starts within 24 hours. It kind of starts. It'll be soft again, but 3 to 4 hours after death, rigor mortis sets in, and that is because ATP is no longer being synthesized. Our cells are dead, so they're no longer using glucose, breaking glucose down and making more ATP. So if you don't have any ATP, then you can't unattach the myosin, you can't detach the myosin from the actin. So the myosin head stays bound to the actin, the muscles stay contracted, and that makes them stiff. And then the muscle proteins eventually break down, and that will cause the release of myosin, and the body won't be stiff anymore. Okay, so we have some more information in Chapter 9. I think I've covered the hardest information, so I'm not going to make any lectures for Part B and Part C, but if you do have questions, please let me know. So these are the lectures only for Part A, but that is the hardest part to understand. So hopefully, if you have any questions in Part B or Part C, please let me know. I can always make a little video explaining some of the tougher concepts.